Good afternoon or good evening to everyone. My name is Gretchie C. Gara. I am from National University, Philippines. And today I'll be presenting the results of the study entitled Beta-glucosidase activity in soil containing Fusarium Oxysporum Cubense planted with cover crops in greenhouse and field settings. The Philippines remains one of the world's leading producers of Cavendish banana and is the top exporter in Asia. So if you try to look at the data that I am presenting, we can see that the Philippines is really one of the leading producers of Cavendish banana. But despite its economic significance, Cavendish banana production in the country faces increasing threats from destructive plant diseases. And one of the most severe challenges is Fusarium Oxysporum Cubense tropical race 4 or FOC-TR4. It is a highly persistent soil-borne pathogen responsible for Panama disease or Fusarium wilt. So what is this FOC-TR4? So this disease invades the plant's vascular system, leading to yellowing, wilting, and eventually death, and can survive in the soil not just for days, not just for months, but for decades. Due to its resilience, FOC-TR4 has proven extremely difficult to manage, resulting in declining production areas and forcing many farmers to abandon infected fields or shift to alternative crops or use chemicals in dealing with this type of challenge. So banana farms in the Philippines use various chemicals in controlling the spread of Fusarium wilt. However, we all know that chemicals may not only harm the environment, but it also harm the farmers and the community surrounding the area. Thus, this study opens a conventional way of eradicating FOC-TR4, and that is by incorporating biological controls such as introduction of microorganisms and cover crops that are found to lessen the threat of FOC-TR4. Cover crops help in enhancing soil health by increasing microbial activity and increasing competition among microorganisms. It also helps in enhancing soil health by providing nutrients to microorganisms. Now, for the objectives of this study uh, general objective is to determine beta-glucosidase enzyme activity using microgram of p-nitrophenol release in grams per soil per hour in soil planted with cover crops, namely Arachis pintoi, Calapogonium mucunoides, Ipomea batatas, and Axonopus comprexus in the greenhouse setting. The second general objective is to determine beta-glucosidase enzyme activity using micrograms of p-nitrophenol release in grams per soil per hour in soil planted with Arachis pintoi and inoculated with biocontrolled in the field setting. Now, why we need to measure beta-glucosidase activity? So, beta-glucosidase is used to measure the effect of cover crops on lessening Fusarium wilt in banana farms because it serves as a key indicator of soil microbial activity and organic matter decomposition. So when cover crops are introduced, they stimulate beneficial microbial communities that compete with or suppress Fusarium oxysporum. And through that, nutrient competition production of antifungal compounds can be triggered. So thus, higher beta-glucosidase activity in soils under cover crops indicate a more active and resilient microbial environment which can naturally reduce the prevalence and severity of Fusarium wilt while promoting sustainable banana farming practices. So for the materials and methods, the study was conducted in two different settings. So we have the greenhouse setting or the greenhouse pot experiment that was conducted at the University of Southeastern Philippines in Davao City, and the field setting experiment was conducted at Puyod Banana Farm, Lasang, Davao City. Now, for the pot experiment, it was carried in a greenhouse setting, and pots were prepared or treatments were prepared. A total of 15 treatments with four controls were prepared in the greenhouse pot experiment. Now, these treatments were actually divided into into four. So we have treatments that are singly planted with cover crops. There are also treatments with multiple cover crops, planted in combination of 2, planted in combination of 3, and planted in combination of 4. Controls were also established in this experiment. Then after treatment preparation, soil were collected. Now for the field experiment, sampling area was laid out using randomized complete block design, and the treatments were also prepared. So the cover crop that was used in the field setting was Arachis pintoi, and it is also introduced or inoculated with a biocontrol such as Trichoderma zenium. So this is a species of bacteria. And after treatment preparation, the cover crops were collected for biomass measurement, and the soil was also collected. Now, these are the cover crops that were used. We have the Arachis pintoi or the pinto peanut. We have Ipomea batatas or the ornamental sweet potato, the kudso or the sea mucunoides, and the acomprexos or the green fraggrass. So after soil collection, the soil was analyzed in the laboratory, and proper decontamination was conducted. Now, for the statistical analysis, so the greenhouse pot experiment used chaparral wilt and levines, ANOVA and Tukis. And for the Puyod Farm field experiment, we've used chaparral wilt, Cruscalwalis, one-way ANOVA, post hoc using paired t-test and Pearson-R correlation test. Now, let's move forward to the results and discussion. For the greenhouse experiment, results show high beta-glucosidase activity in single planted and in multiple combinations of cover crops. So we have here significant difference between 4 treatments and 1 control. So treatment 3 with ornamental sweet potato, treatment 5 with pinto peanut, treatment 11 with pinto peanut, kudso, and ornamental sweet potato, and treatment 14 with kudso, ornamental sweet potato, and green fraggrass shows significant difference with that of the control group, which is bear with grand nane and inoculated with Fusarium. So what does this result show? So this shows that there is an effect of planting cover crops in Fusarium infected soil. So Ipomea batatas has shown to be an effective cover crop in increasing the amount of enzymatic activity, whether it's planted singly or in combination with other cover crops. Leguminous cover crops such as Arachis pintoi and C. mucunoides were also found out to be effective cover crops in increasing the amount of enzymatic activity planted in combination with non-leguminous cover crops. So in the development of the greenhouse pot experiment, Ipomea batatas or the ornamental sweet potato observed to grow faster and easier compared with the other cover crops that were used in the experiment. It also gave higher organic matter since it has broader and more numerous leaves than other cover crops. So the study conducted by Nidut Sikizan showed that sweet potato has the ability to produce higher dry matter per unit area, per unit time, that may induce high level of enzyme activity in the soil. Arachis pintoi or pinto peanut also showed outstanding effect in increasing the amount of organic matter in the soil. Arachis pintoi or pinto peanut basically has small but numerous leaves and can also thrive easily in the soil. So according to the study of Radovic, Arachis pintoi produces dense cover with minimal maintenance and may protect soil from erosion. It is also an appealing ground cover in agricultural areas because of its ability to fix nitrogen. It is also observed with greater cover crop in terms of mineral production and had significantly enhanced the availability of carbon and nitrogen in the soil. Now for the Puyod field experiment, it shows that there's no significant difference among treatments, but all treatments showed high enzymatic activity. Now, since there's no significant difference among treatments, we used effect size to get the relationships between time of collection. And during different collection time, it shows that collection 3 and collection 2 has largest observed difference, followed by collection 1 and collection 2 with moderate to large difference, and collection 1 and collection 3 with small difference. So this bar chart here visually supports the moderate to large effect size between collection 2 and 3 and collection 1 and 2. So this reinforces the idea that while mean comparisons may appear similar, the underlying variability explained by time of collection is meaningful as reflected by the effect size. Now, since there's a relationship between collection time, we test the significant difference regardless of the treatment. So this table shows that the first collection, okay, and the third collection is significantly different from that of the second collection. So the first collection was conducted in season with high precipitation, as well as the third collection. And however, the second collection was conducted in the time with low precipitation being recorded. So these findings indicate that precipitation had great influence in soil moisture content, enhancing microbial activity in the soil. According to Su in 2020, higher levels of soil moisture resulting from increased precipitation not only enhanced biomass production but also led to greater residue accumulation, providing an essential food source for soil microorganisms and contributing to improved soil health over time. Now, for the summary, conclusion, and recommendation of this study, so for the greenhouse setting, there's high beta-glucosidase activity that was observed in single and in multiple combination of cover crops. Ipomea batatas, Arachis pintoi, and C. mucunoides were observed to enhance beta-glucosidase activity in the soil. For the Puyod farm field experiment, it was found out that there's no significant difference among treatments but showed significant difference in the time of collection. The results suggest that either ground cover or biocontrol can be a good source of beta-glucosidase activity in the soil, and the factors that affect the enzyme activity were temperature, period of planting, and the choice of cover crops to be planted in the field. Now, it's recommended that the duration of the study should be extended. Maybe it should be extended for for 3 years or 5 years. Related studies on this using the same species of cover crops should be conducted in other farms to compare the significant effect of ground covers in enhancing the main crop yield, and further studies should monitor the growth of cover crops every week to avoid damage in cover crops, since seasons are changing, the weather is continually changing, every day it's changing, and try other types of cover crops that can stand in a harsh and varying environmental condition in the field experiment. So these are the references that I've used. I hope that you learned something from the study that we had. Thank you so much for listening. Magandang araw po sa inyong lahat.